Meiosis. In this video series, you are going to learn how things are inherited, how there's variation when things are inherited, how do we get new genetic information. In order for meiosis to happen, you have to start off with a regular old cell. So a cell that's 2N. Meiosis is going to take place in your gonads. So in females, we're in the ovaries, and in males, we're in the testes. And these organs are making sex cells. And inside of those cells, those organs, we have a full copy of genetic information that represents you. It's the same genetic information copies that are in every other cell in your body. So we start off with a diploid cell. Then they split in half to make N, right? So we duplicate chromosomes to split in half, and then in meiosis 2, we unduplicate them to make 4 different cells. So every time we go through a round of meiosis, one cell ultimately turns into 4 cells. So from one cell, we can make 4 egg cells or 4 sperm cells. Remember I said that you have 23 pairs? Like that one's from mom, that one's from dad, that one's from dad, that one's from mom. Those are what we call homologous chromosome, homologous pairs. They're dad's number 1 chromosome and mom's number 1 chromosome. So they're alike. They have the same genetic stuff that codes for the same type of protein, but it could be different in mom than it is in dad. Like mom's genetic code could be for brown eyes, but dad's could be for blue eyes. But you have both copies. So we call those homologous chromosomes. Those are pairs that have the same types of genes on them. And you remember you got one from mom and one from dad. But those are different from sister chromatids, which are 2 identical copies of the exact same chromosome. So if this is mom's number 6 chromosome, if I make 2 identical copies of it, these guys are then sister chromatids. So the purpose of meiosis is to produce these sex cells, these gametes. It's the process of creating them. Sex cells are going to have half of your normal genetic information, and they're only going to have 1 set of chromosomes. So they don't exist on their own. They need to find another copy of DNA to go through mitosis with. To do this, cell division happens twice. It happens in round 1, meiosis, which is where we separate the homologous chromosomes. Okay? Those are the homologs. There was a set you got from mom and a set you got from dad, but it's not like all of what will eventually be grandma's cells, chromosomes, go into one cell versus the other. They're going to be all mixed up, okay? And in meiosis 1, we see a reduction from that diploid cell, or 2N, to a haploid cell, making duplicated N chromosomes. In meiosis 2, that's when we're going to separate the sister chromatids. So sister chromatids are now like identical chromosomes that we're going to split in half again. Meiosis has 2 rounds of separation, if you will. This meiosis 2, duplicated chromosomes from meiosis 1 will then divide into individual chromosomes. So look at the diagram. Here's the gamete cell we're going to start off with. All right. Here are 2 chromosomes, pairs. So in this cell, there's 1 copy that this person got from mom. There we got 1 from dad. That was from dad. That was from mom. Okay. But this individual is now going to produce gametes to make their own baby. So these chromosomes, this is what is existing in your normal body, right? Like we have 23 of these guys, 23 pairs. All right, but 46 individuals. So those guys in meiosis 1 are going to duplicate themselves. They're going to go through a process of duplication, all right, and then separation. What we see where it kind of exchanges these little colors right here, that's called crossing over, and we'll talk about that here in a little bit. But when they get really close to each other right here, and when in metaphase, when they get really, really close to each other and they line up in the middle, they can switch DNA. We'll talk more about that in a minute. Okay, so then these duplicated pairs are going to get pulled to one side, all right? And then we're going to form two separate cells, two separate potential gamete cells, and each of these cells is still 2N. It has duplicated DNA in it, all right? And this one's 2N and that one's 2N. Now these cells right here these guys right here, these chromosomes are homologous pairs, all right? So in meiosis 1, we're separating out homologous pairs. Now we have these duplicated chromosomes. Those are called sister chromatids. They're sister chromatids. They're unique to meiosis. They're really unique to meiosis 2. All right, so sister chromatids, you could see that these guys, these two cells right here, they're identical. This is an identical cell, and that's an identical cell. See, we duplicated it, and then created like two identical type of cells, really similar to regular cell division. All right, but now we're going to take these two identical cells and like split them in half. And so because this cell and this cell, their chromatids are identical, they're called sister chromatids. And in meiosis 2, we're going to separate the sister chromatids. So these duplicated chromosomes in meiosis 2 are from a result of meiosis 1, and they're going to divide into individualized chromosomes. So now we're going to split them in half, creating 4 cells down here at the bottom that have different versions of DNA that were on the original chromosomes. So our cells are doing this to randomize genetic information. You can think of it as, if you know your biological grandparents, you favor in how they look a little bit maybe. You don't look exactly like them, nor do you look exactly like your biological parents, but you favor them. So there are things that you kind of have got from mom or dad and kind of look similar to, your grandparents. And because there's been this randomization, this mixing up and exchanging of DNA, you have the right amount of DNA. It's just that certain pieces of it came from different family members. All right, so before meiosis 1 can even happen, we have cellular division, right? Interphase, that's that growth cycle, that G1S, G2 phase. It happens in 3 parts. This G1 phase, which is gap 1, and that's where the cell grows and it makes proteins. It's just like doing like normal cell stuff. It's just like cell stuff, right? And then S phase, that's in synthesis. That's when it goes ahead to duplicate its DNA and doubles the number of chromosomes that it's going to normally have. And then in G2, since it has all this like duplicated genetic material, then it's going to go through gap 2 phase, and it's going to grow and like do more protein synthesis. And at the end of interphase, the cell has 2 sets of duplicated chromosomes, and that's where mitosis would take over and divide. But in this video, we are in sex cells. We're going to make gametes. These are very specialized cells that happen only in the gonads of sexually reproductive creatures. Okay? So going through before meiosis 1 even happens, this normal cell cycle of interphase, which was 3 parts, happens inside of these cells that will eventually be gametes. S phase is so important. Unduplicated chromosomes will go through DNA replication during that S phase of interphase, and they will become duplicated because they're an identical duplication of themselves, technically, of each of these things. We call those homologous chromosomes. Now, this is not a normal cell, right? Normal cells don't have copies of multiple copies like this. This cell only has duplicated homologous chromosomes because it's getting ready to divide. So it needed to duplicate its DNA so that the new cell would have all the right genetic information. But these guys that duplicated, that are identical, are the sister chromatids. So the chromosome is this like X-like structure, and the chromatid is the single part like that.